My name is Michael Dickens, and I'm a research scientist with HPRC. The course today is Intro to Next Generation Sequencing Data Analysis. So we'll go over some cluster utilities and then help you get familiar with the cluster. And then we'll go over some next generation sequencing technology, some sequencers, how they sequence, and then we'll show you how to find tools on Grace. And then we have some template job scripts that already have sample input files configured, so you can just run them to see how they work. And you can use them for your own research. All you got to do is change the input files and maybe add more wall time and more cores. And maybe more options that aren't in the template script, options for the software command. And then we're gonna run 4 jobs. The first one, we're gonna look at some raw sequence data, look at the quality of the raw sequence data. Then we're going to trim the reads, and then we're going to compare before and after trimming. And then we'll align the reads to a reference genome, and then we'll run it on sequence calling some variants, and you'll be able to find SNPs and indels. And if we have time, we'll cover biocontainers. So there's a number of ways you can run bioinformatic tools. We have our portal. So the portal has different ways to access the cluster. You can use these interactive apps such as JBrowse, or even might use VNC. VNC basically launches a job on the compute node, and you get a terminal, and then you can work on the terminal on the compute node interactively. But if you're not going to be working interactively, then you just submit a job script, submit it, and it runs and while you're away. Running these applications on the portal does start a job, so it charges service units. So you have to have service units, which you have to renew each fiscal year, which ends August 30th, September 30th, one of those two. Sandra, when does fiscal year end? September 30th or August 30th?
August 31st.
August 31st, okay. So September 1st, by then you need to renew your account if you haven't already. You can use the Unix command line. You do need to learn Unix and Slurm, but it's very useful. This is the way you're going to be, you're going to have more experience. He'll take more experience away if you use these from you as you move on. If you learn the Unix if you learn to use the Unix command line and learn about Slurm, because the portal's good, but everything's done in the background. If you go somewhere else and they don't have a portal, then you might have to use the command line. We also have Maroon Galaxy. It's a Galaxy installation on Grace. There's a lot of tools available. It's all web browser based. It's all click. You don't have to know any commands. All the tools that are on the command line or basically have a web interface in Galaxy. So all you do is make your selections, input files, options. And if you want to use Galaxy, first you get your HPRC account, then you request a Galaxy account. All right, so let's connect to, we were going to use Grace, but does everyone here have an access ID? Did you get an access ID? Okay, so we're going to use Launch instead. It'll be a lot quicker. Grace is having some issues this week. So instead of going to Grace, we're going to go to Launch. So from here, you can go Portal, Launch, and log in. And then do your 2-factor authentication. And it's a phone call. Everyone got on launch? I got on launch. Did you get on launch? Not yet. Okay, we'll wait just a minute.
Let's add to the application.
Has she been added?
No.
Oh.
Let's ask it again there for West Center.
Okay. But you can still get on to launch, right? Without being on the allocation.
Oh, yes, yes.
Yeah, we'll get to running jobs in a little bit. By that time, you'll probably be added. So just go ahead and log into launch and let us know when you're logged in. And you go to our main website and then portal launch. So launch has some file software installed. Grace has the most. Well, actually, Faster has the most because we had to reinstall a lot of Grace software modules. But Grace has a lot. Faster has a lot. Okay, if you're not familiar with HPC, how it's set up, generally we have the login nodes. This is where you do file manipulation, create your text files, upload, download files. You can install software, do some light testing. Any jobs or any tasks that are running more than 60 minutes. Once they reach 60 minutes, we have a system script that will stop the process. So that way, not everyone's using the login nodes, all the resources on the login nodes. And you also want to limit it to 8 cores. We also have some scripts that will cancel your commands if they're using more than 8 cores. And if they're using too much memory. You don't get charged service units for logging into the command line. That's where you do file manipulation, creating text files, and submitting job scripts, reviewing output files. But when you do have a computational job that requires lots of cores, lots of memory, then you use the Slurm job scheduler. You'll create a job script, we'll go over that, and then you'll submit it to Slurm, and Slurm will look at all the compute nodes and find the resources meaning available cores and memory. When it finds a node that it can fit your job on, your job will start running on that node. And then when it's complete, it will. Well, when it's running, it'll show. Well, when you first submit it, it'll show up in the job queue as pending. And then when resources are found, it'll show as running. And then when it's completed for like less than a minute, it'll show as completed. And then it'll go away from the job queue. So if you search the queue and your job's not there anymore, that means it's completed. Either it failed, stopped early, or it completed running successfully. And we'll show you how to see whether it ran successfully or failed. These jobs, the Grace, well, let's see. We're going to be on launch, but Grace has 48 cores and 360 gigabytes of memory, and I'll show you how to figure out the maximum cores and memory on a compute node. And anything running close to 60 minutes or more, just run it as a job script. And you also get an idea of how much memory and how many cores it's using. And when you do submit jobs, it does charge service units. Okay, we're going to go over some cluster utilities. We have a number of utilities that help you understand the cluster. With any of these, you can type in the command and -H or -help, one or the other, and it will show you usage on how to use the utility. So the first one, well, let's skip that one. Well, I'll just go over. We can go over it later again too when we run our jobs. So you get a job ID each time you submit a job. You don't know what the job ID is yet. Slurm has a counter that goes from job 1. In this case, this is job number 11,760,808. So whoever submits a job after this is going to be 11,760,809. So it goes sequentially, but you don't know what your number is going to be till the job launches. So once you do get your job ID, then you can run this my job and then the job ID, and it shows you some information. In this case, it completed successfully. Exit code is 0, means there's no errors. If you see exit code 1 or some other number, that means there was some kind of error, either from the software or you ran out of memory and Slurm canceled the job. It shows you the compute node, the time started, time submitted, time started. So whenever I submitted this, it started about 21 seconds later. It ran for 7 hours, 30 minutes, 28 seconds. It used 37 gigabytes. I requested 180, so it used 20% of the memory. It used very low CPU efficiency. That might be because it depends on the software. Oh, this is AlphaFold. It could be because it was using GPUs and it was using a lot of system, their motherboard CPUs. It also shows you some other information, where you submitted it from and the name of your job script. There's also a way to, if you run this with a -S after a space -S at the end, it will show you your job script. It saves a copy of your job script in case you deleted it or changed it and can't remember what you had in your job script. So run -S, and at the bottom of this, it'll print out the job script. We'll do that later. It also shows you if your job is pending due to maintenance. In this case, if your job's been pending for like 6 hours, you're not sure, then you can run my job and it'll say there's a maintenance that starts at 22 hours. So your job will not launch until after the maintenance is complete. That's if your job, the end time overlaps with the start time of the maintenance. Here's another one. If you request too much time for the partition, your job could remain pending and you don't know why it's pending. If you run this, it'll tell you there's a partition time limit of 2 days, but you requested 7 days. So you need to cancel this job and change your SBatch parameters for 2 days. All right, max config. This is on my job's on all of our clusters, so is max config. This one shows you the maximum cores and memory and hours and nodes you can select on a compute node when you want to schedule a job. Let's just say you're running an alignment, VWA alignment. VWA can take advantage of all the cores. So if you run it with one core, it's going to take a long time. You might think, I'm going to save service units and just use one core, but you're actually it's going to take so long where you'll probably be using more service units. Just use all the cores and it'll get done a lot quicker. And the way you figure that out is you run max config. So were you able to get on a cluster on launch? Not yet. You are on launch or Grace? Okay, so you can get on launch. Okay, so you can go to our main web page and go to portal launch. It's hprc.tamu.edu and then mouse over portal and you'll see launch. Oh, so when you registered, you probably used, did you use Texas A&M or have you had an Actus ID for a long time? Do you remember what you used? Did you just get it this week?
Last week.
Oh, okay. You can try with this, and if it doesn't find you, then you can select Texas A&M. You can do Texas A&M first. Just type in Texas A&M and it'll find it. Yeah, just scroll up. Yeah, select that and then do next. Don't go log on yet. And then you type in, go back, select portal. Andrew, you can post this. See if they fix it. Failed to launch, failed to map, MALON. Okay, go back to look for launching. Okay, trying to open a private web browser or incognito tab. And if you will go off here, please. Or you can go right here. Do incognito. Okay. Now do HPRC.tv. And then do portal launch. And then this time, go ahead and try access. Try that one. Yeah, it's the first one. I'm sure you did A now, but see if you can log in. Do you remember your access ID and password?
It's already on that.
You can find the oh, you're already on launch. Okay. I think maybe you did do use the years. Just close that and close that. Oh, that yeah. And open a new one. Here. And this time, go to HPRC again and launch. And select text layer. And with your ANAM tiny pass. And then I do the password. It won't be your email, it's just the first part of your email. Do that, fix that, got that at viewer. Thank you. M-E-L-O-M. Oh, M-A-L-O-M.
No, I didn't know that.
Oh, man. You post that maybe West. M-E-L-O. Okay, we're gonna work on that. She's gonna see if we can fix that. Okay, so this is something you can run on the terminal. So you can go to cluster, launch shell. Okay. And just run max config. So by default, it will show you the okay, by default, it will show you the maximum parameters for a CPU node for the non-GPU node. So it's showing you the partition CPU, showing you can run for 2 days. That's the maximum time for this CPU node. If you have software that supports multi-cores, you can use up to 4 nodes, or multi-node if software supports multi-nodes. Each of these nodes has 192 cores on it. and 371 gigs memory. So if you want to, you can copy and paste this into your job script and just run it that way. Notice I have two output files, output and error. Output, there's going to be a file called standard out and another one called standard error. It's going to have %x takes the job name. So the output file will be standard out dot my job dot %j is the job ID. You don't know what that is yet, but when it runs, it will fill it in. So you're going to have an output file with the job ID and the name, and that helps you identify which job you ran, and you can help debug it if you need to. If you want to check the GPUs, you can type in the type of GPU and the number. But since there's not A100s on, so you need to use A30. So GA30. If you do GA30, it'll show you the maximum is 2. You can specify just one GPU if you do -G1. It shows you, notice there's a couple different added lines here. Now you can do, now the SBATC partition line is in there, that's required. But it also adds this line in there for you. So if you can use a GPU job, if your software supports GPUs and it supports multi-GPUs, you can use that, copy and paste that. Notice the amount of memory is twice as much on the GPU node as it is the CPU node. GPU avail, that shows you which GPUs are available, how many we have installed, and how many are available for jobs. You can see all 10 right now are available. 2 are reserved. We have 10 nodes. Each node has 2 A30s. These are all 10. So nobody's using these right now. You can also access these utilities from the dashboard. You go to Utilities, GPU Avail. You'll see the same output. You don't see the reserve nodes here. I need to update this so it shows the reserve nodes. Okay. CPU Avail, that can be useful in these smaller clusters. Just run CPU Avail, Control L to clear. You can see that there's a group of nodes here that have all the memory and core available. So some of them are only partially available. There's some more in there. So there's plenty of nodes if you need to use all the memories and all the cores. Maintenance, just run that. It'll let you know if there's a maintenance coming up. Right now, it says there's no maintenance scheduled. All right. We're going to try to add these shortcuts. They'll be useful in the future when we run some jobs. So we're going to have a lot of files. So one thing is when you do LS, I have very few files here, but when you start to work on a project, you're going to have like 50 files maybe. And when you run something new and you do LS, it's sorted alphabetically. You can see this is sorted alphabetically. So you can't really tell what was just created or what's being created. So if you create an alias, you can do ls-lsth, okay, and it will show you the newest files on top. You can see the newest file was created September 8th and goes all the way back to January of 2014. Okay, so let's add this to our bash RC. We'll add the first one here, and then so let's come to here, go to Files, go to the portal, go to Files and Home Directory. You can still do this. We're not running jobs yet. Oh, you can't even log in. That's right. She's having trouble mapping. I don't know what that error is. Okay. Yeah, maybe it will. So you can close your incognito browser tab and then start a new one and try to go through it again. It might be ready now. Okay, so we selected home. You should be in home and then your user ID. Is that where you are right now? Okay. We don't see a .bash RC, but anything that starts with a dot is hidden. So you need to click this show dot files. Now look, there's a lot of files in there you didn't even know were there. Okay, here's bash RC. So what we're going to do is we're going to click here and we're going to do edit. Okay, see if I can make this bigger. Okay, I've already got this in my, basically you want to add a line that looks like this. Line 20. You can call it whatever you want, but I just do LHL. It might be good just to use that for now. So alias space LHL equals and use quotes. You can add the others later, but this is the best, most important one. I'll also add line 19. It doesn't matter which order you add this in. So let's add line 19 and 20. These two. So basically this just says whenever I type in Q, instead of having to type in all of this, I just hit Q and hit enter. It's like a shortcut. Okay, how many people are finished? Okay, make sure you click save. Top left corner. It's probably dark blue if you have some edits made, and when you click it, it goes to light blue. And then once you save it, you can quit, or you can close this tab. Did you try again? Still the same? Did you get yours set up? Okay, so then when you're done, you can save it. Make sure you save it, and then close that. Now, from here, you need to type in this command. Source.bashrc from your home file. Okay. That way it reads that bash RC again. And then you can do LHL. I don't know if you'll have any, you should have two directories at least. So did everyone get LHL to work? And then you don't have to source it next time you log in. We're just source it this first time because you've already got a shell open. When you leave and you start a session again, you don't have to source again because when you open a session, it automatically reads your bash RC. But we needed to reread it because we just made an edit. Okay, how many people successfully got that done? You need help? Maybe. I see it's command run. Okay, did you run source? Yes, that's fine. Let's see your you need to take space out right there. Oh, look, wait. Oh no, that's you put it in a different corner. No, you put it in the file. You need to put it in this file right here. Yeah, I'm telling you. Let's see. Okay, spell alias, A-L-A-I-S. A-L.
Sorry.
I-A-S. You got it? Okay. So, that's it. Now you go to cluster, long show axis. Okay, go back here. You type in yes. Right here. Type in yes. Enter. Okay. And go to files. Directory. But show the dot files. Come down and click this. And head it. And then add these two lines. Add these two lines. 19 and 20. You see that? All right. 19 and 20. So did you get ADS to work? No, this is ADS is spelled. We fixed it. Yeah. I've been alias Q equals just like that. Like that. And they orange from the sequence. It's SQUEU. It's kind of really it's different. And anything was it here. I can source SOURC space dot VL bash RC arsenal and so and dot Q clear okay, own jobs. So that command. Okay. Cool. Okay, so now I'm going to quickly cover sequencing technologies. Illumina has a lot of sequencers, from a very small one that's about the size of a printer to these taller ones. The small ones, you can do the smaller genomes like bacterial genomes. We have single end and paired end reads and mate pair reads. Single end, the DNA is fragmented, same as paired end. But with single end and paired end, you get these like adapters that are ligated on each end of the double-stranded DNA. And then the sequencing is done. For single end, it only reads one of the, it only sequence one strand. Double-stranded paired end sequencing will sequence both strands. It depends on what analysis you're doing. You may only need single end to get the data you need. You can imagine single end doesn't cost as much as paired end because there's more sequencing involved in paired end. Mate pairs, this is creating a long insert, sometimes 2,000, sometimes 10,000, that helps with genome assembly. So let's just take an example, double-stranded DNA. If the entire fragment is 400 base pairs long and your sequence read is only 100, then you're going to sequence the top strand 100, you're going to sequence the bottom strand 100, and there's 200 bases in the middle. You don't know what that is. But you're going to get enough fragments that when you do an alignment, and they'll overlap, and you'll be able to find out what's in the middle there. You'll get two FastQ. You get two files. Read one FastQ and read two FastQ. This is the format. Notice it has the headers the same, except for the one here and the two here. That way you know this is read two. You should have the same number of lines in both these files because this is like, if this is the first entry, this is one fragment of double-stranded fragment. That's the top strand, that's the bottom strand. Okay, sometimes you do have to maintain the order so that the number of lines will be the same. Sometimes like if you look at fragment 3, the left and the right, they overlap a little bit. The second one, there's a bigger gap in the middle. So there's different fragment lengths. So you don't know what's in the middle. Some of those you don't know, but some they overlap. Adapters, we do some adapter trimming, and the reason for that is because whenever these sequence adapters are ligated for the sequencing process, they're on the ends of the strands. And then let's just say the sequencing is longer than the fragment. So the sequencing on the top strand starts here, it goes across, and it reads all the way to the end. It doesn't know when to stop. It just reads to the adapter on the end here. So we need to trim that off because it's not part of the DNA. It's not part of the genome. So we'll do some trimming in a little bit. Same way with the bottom. It starts at the bottom. It can read all the way to the end. But if the fragment's really long, it doesn't read the adapter. So some of them will have adapters. A lot of them will not. The Oxford Nanopore, this is longer reads. Has a different way to sequence than Illumina. They have these little units. They're about as big as a laptop, and then they have like the USB unit where the actual sequencing goes on. And these are the ones that are portable enough where they can sequence in space or out in the field. PacBio, this is like a refrigerator. It's pretty big, but you can get long reads, 20,000 base pairs as opposed to 300 base pairs. The accuracy is not as high, but it can get very high for these shorter reads, because what it does, it creates a loop like this by putting these templates on the end. It'll sequence the top and bottom strand until the reagents wear out, and you'll get this one long read right here. So you'll get the top strand sequence multiple times and the bottom strand sequence multiple times. So you can imagine a shorter read, the reagents are going to go a long time. You're going to get a lot of sequencing on the top and bottom, but a very long read, you may only go across once or twice. So then you can create kind of like a consensus sequence, and the more reads you have, the more accurate it is. So the shorter PacBios are more accurate than the very long ones, and it can sequence very long, like 50,000 base pairs. You can see these hi-fi reads are more accurate and they're shorter, opposed to the very long reads, which are not as accurate, but they're very long. They can still be used for scaffolding or something else. So is everyone familiar with searching for tools using Module Spider? What about you? You familiar with Module Spider?
I'm just starting confused.
Okay. Okay. What about you? Use Module Spider?
Yes.
Okay. So we can do this on the command line. So just type in Module Spider and then the tool name. We'll just type in SAMTools. Module Spider SAMTools. There's now notice the output here. We see it matches SAMTools, but it's our SAMTools, which is different than the SAMTools we're looking for. But look down here, it says it's showing lines 1 to 14. So we need to see more. It's only showing the first page. So you hit spacebar and it shows you more. Now this is what we're looking for, SAMTools. Notice we have a lot of versions. You may want to use the latest version. Now, if you don't want to see, if you found what you want, you don't want to see anything else, you can hit Q and it'll quit. It won't keep paging through. Okay. So the way we did that was Module Spider SAMTools. Now it wasn't case sensitive, but if there's an exact match, it will be case sensitive. So if I type it in, it's going to show me an exact match. Okay. And Q to quit. Okay. You can also use module key and type in the word assembly. It'll search the description. If you're doing genome assembly and you want to search for what modules are available, module key assembly. Any questions on that? Module Spider. Okay, that doesn't mean you loaded anything. That just means you're looking for software. Here's some other tools that a lot of users are using. Minimap 2, this is for PacBio sequencing. Canoe, Unicycler, and WT-DBG2 for assembly. Fly is also still being used. Improved draft assemblies, there's a couple of tools we have. If they're not installed now, we can reinstall them. There's also a tool suite for PacBio called SmartLink. There's a lot of utilities that are developed by PacBio or for PacBio. Hybrid assembly means you're taking these long reads and the Illumina short reads and you're creating assembly. Spades, Mazurka, and Unicycler can do that. Big genomes use Mazurka, smaller genomes use Unicycler. The bacterial genomes is Unicycler, and then smaller genomes, maybe fungal genomes, you can use Spades. So we're going to do a little exercise here to see how to load modules. So you want to, first we did Module Spider SAMTools. We already did that, but now you need to know how to load it. So we're going to do Module Spider and we're going to type or copy and paste the name of the module. So type in Module Spider, and I'm going to highlight this, and notice whenever I highlight it, you see that little copy thing that popped up? That means it copied it into my clipboard. So highlight this and then right click and it will paste it for you. Yeah.
Yes, so no, I don't have the 1.2. It's 1.82.
You're on launch? Did it get cut off?
Just find it. No.
Oh yeah, yeah, it got cut off. You need to you just type in 20. Yeah. Enter. Yeah, I think it got cut off when it was displaying. Okay.
Oh, okay.
So Module Spider. Okay, did everyone get this? If you didn't get it yet, raise your hand. You need me to redo. I'm going to hit Q to quit because I've already got the information I need. Q to quit. So I did Module Spider and I copied and pasted case sensitive name of the module, SAMTools 1.20. Did you get it?
Yes.
Okay. Right, you. Let's look at it. I need the space. Okay. Now we got this information. It tells us you need to load all modules on any one of the lines below. This is a software tool chain that was used to compile this software. You might see GCC 12.3.0, GCC 13.3.0, GCC 11.2.0, something like that. You might see multiple lines. In this case, there's only one line. If there are multiple lines, you only need to pick one of those lines. So we're going to load this module. So module, load, and right click. Okay. And then space. Then we can load SAMTools. Right click. So this is what your command should look like. And then you can enter. And there's no messages. If there's no error messages or no messages about things being reloaded, then everything's good. Sometimes it says module doesn't exist. That means you either typed it in wrong or it doesn't exist. If you typed it in wrong, then it doesn't exist. So did everyone get this module load? Okay, you can do module list. Terminals, non-function flaggers. I don't know what happened there. Module list. And it shows you what was the modules that were loaded. Did y'all get this terminal not functional error? I don't know what happened there. Module list. Did you get module loaded? You got modules loaded? Okay. Ready?
You would make each one list.
You need to go over one. You need to space. Okay, now do module list. So notice we only loaded 2 modules, but there's 10 loaded. That loads all the dependencies for you. Okay. Now we want to see what BWA module we can load, a module that's compatible with the same tool chain. So you can do module avail. It shows you which BWA modules are available. So let's type in this one, module avail BWA. Module avail BWA. Okay, I don't know what that fully functions. Okay, 13.3. Oh, actually, we don't have one. We don't have a BWA module for 13.3. See this? It only goes up to 12.3. Okay. But if there was one available, you could do it that way. You could load it also. Even SAMTools, we only had one 13.3. Okay. Then let's do module avail bowtie 2. No, there's not one. Bowtie 1. Just do module avail. Module avail. It shows you the modules that are available for 13.3.0. You can see you can load any of these if you want. There's a lot more. But we don't have one for BWA. Okay, so then when you're done, you can do module purge. Module list. Module purge. Module list again, no modules loaded. Okay, I'm going to close this. So go ahead and close your tab and let's start a new session. Close that and let's start. Go to the portal again and start a new shell session. I've never seen that error before. Hmm. Oh, I think we did need to do 1.8. Okay, so this one has 12.3. See, we needed to load 1.18. 1.18 loads 12.3, and then you can also load BWA with 12.3. Okay. We're going to see more of that in a little bit. Okay, temp directory. So whenever you're running, if you know we have quotas, so you don't want to create 5 terabytes of data and 100 million files. There's a temp directory on the compute node. It's an NVMe drive, it's like a flash drive. You can write intermediate files there, and it doesn't count against your quota. And it's also going to be a lot quicker because it's directly on the compute node. You don't have to write across the network. So if you can specify in your command somehow, if you can specify a temp directory, like for example, this is the card, you can actually specify a temp directory. Then you can do it this way. Now, $TMPDAR, all uppercase, that doesn't exist on the login nodes. It only gets created when the job is created. It's temporarily there. You use it when your job is running, and when your job completes, that directory gets deleted. So it is a temporary directory. All right, so quality evaluation, we want to see what the sequence reads look like that come off a sequencer. FastQC is one of the most used software to evaluate sequence quality. It only reads like 200,000 reads. It gets an idea of the quality. It doesn't have to read all 5 gigabytes or however much memory there is. So we'll do that in a little bit. But there's if you ever hear about normalization, digital normalization. So whenever you're doing some sort of assembly or some analysis, the assembler might say, don't use more than 200x because it can cause issues with the graphs, or it could just take too long. And let's say you have 1,000x because you sequence a small genome a lot. You don't need 1,000x. So you can reduce the coverage. And there's a tool that will do that called BBMap. You can look into BBMap and you can reduce the coverage without losing, with basically you're reducing redundant coverage. You're not losing a lot of data. Trinity, it now by default will normalize reads to a depth of 50x instead of trying to assemble 200x. It doesn't need 200x, it only needs 50x. So it uses ChIP-seq, that's software to do the normalization. Okay, so we're going to use a utility called GCA templates. GCA templates, these are some template job scripts that have data configured input files, so you can just run it to see how it works. And then when you get more familiar with creating job scripts, you can use these as like an example, and then you can just change the input files. So let's create a directory. Anytime you see the green, that means that's something we need to enter. So we're going to make a directory called scratch NGS_class. Now this is all case sensitive. You need to be scratch uppercase, and notice there's no spaces. The only spaces between the Unix command make directory and the path. So type in make directory scratch NGS_class, and then CD to that same directory, and then run this command GCA templates, and you should see this menu that pops up. I'm gonna wait for everyone to get here at this point. close that. Go to the top of this. Yeah. Off shell X. And then do this, type in these three commands. Now the one on the right, that's gray, so we have a lot more templates than we do on launch, but we're going to use just a few templates on launch. It might not be numbered, just since we're on since we're on launch, it's going to be actually number 1, FastQ file. So instead of it says enter 2, but we like that it's 1, or you can use a search function by typing in S.
So
It's just
like three columns together, like in one I
these are correct commands, yes.
just standard.
Yeah.
Okay, okay.
Good. Yeah, this is great. So we're going to use Grace, but it's slow, so we're doing launch. It looks a little different. So we're going to select number 1. Yeah. Okay, and then type in GCAT and hit the tab. Hit the tab. See how it fills it in for you, and hit enter. Okay, now we all got to the menu. It's going to look a little different than what's on the. Now I'm going to go up arrow, and I'm just going to, instead of typing all that in, I'm going to. Okay. So we want to search for FastQC. You can type in S for search, or you can type in 1. We're going to do QC. There's 2 QCs. We're going to do 1, the number 1. That's quality evaluation. Is that what we're doing? Yeah, QC, quality evaluation 1. Now there's FastQC. You might see different tools or different versions of the same tool. We're going to use FastQC 0.12.1. We're going to use the defaults. That means there are no special options. So basically, I think we hit 1. So I went back home. It's 1, 1, 1, 1. Okay. Now it says copy the script to the current directory. You can type Y for yes. And then there's a reminder there that these are template scripts. You need to read the software manual to see if there's any additional options that you can add. You might find newer versions of FastQC. And if you do, you can change the version in the template script. Okay, did everyone get this so far?
Yeah.
You type in Y for yes and hit enter. Okay. Now we want to do, I'm just going to take a quick look at what the script looks like. The very top, you can see the SBatch commands. We've got it configured. It's only going to take like a minute or 2 minutes to run, but we had 1 hour, which gives plenty of time. It's not going to take an hour, but when you charge service units for an hour, if your job completes in 1 minute, you're going to get 59 minutes worth of service units returned to your account. I've only got 2 tasks because I only have 2 files, so it doesn't require a lot of computing power. Here's the module loading. There's a manual link here. Here's the input files. Here is how many threads we're going to use. In this case, it's going to take this value up here 2. This is an environment variable. The output directory is a current directory, which is a dot. You can use a dot /. Sometimes it might be hard to see the dot. So people overlook it. So I always put a dot /, which is the same as just dot. Here's the actual command. And then there's a citation at the bottom for let us know. Use our clusters in your publications. Let us know. Here's information on about letting us know. And then if you'll want to reference the software because they get funding based on or some of them get funding based on how many users are using their software. All right. So anything with a pound sign is a comment, but for SBatch, you have to put the very first line just like it is, no space and nothing after it. The pound SBatch, that's a comment, but Slurm recognizes it. It says this is a parameter I need to search for resources. You can put, notice how I have comments after it here. That's okay. You can't put a comment on the first line, but the rest is okay. All right. So now from here, you can do cat, type in RU and hit tab. It's going to complete the name for you. What cat does is it prints the contents of the file to the screen. Now, if the file is really big, like 2 gigabytes, you don't want to print it to the screen because it's just going to scroll and scroll. Then you have to hit Control-C and it will eventually stop. But we know this is rather short, so just cat, and you can see similar to what was on the slide. Actually, I don't have a module purge here. I should put a module purge, but input files. This is a sequence with the MySeq. This is a genome candida duplinasis. There's a forward and reverse strand. All right. Now we're going to submit this file. To submit the file to the Slurm schedule, you type in SBatch space and type in RU and hit tab. Okay, SBatch run. Now it says, I don't know if you'll see something like this. All right, now type in type in Q, because we remember, so you don't have to type in SQUE-me, you type in Q. Do you see this? Your job's running. I can see how many people are running. Okay, that's because it's already complete. See, my job completed already. So if I do LHL, so y'all should see these files. Do y'all see these files? Everyone see these? Two people confirmed. Do you see them? Okay. So did you do the SBatch run? Okay, sorry. I'll keep this back up. And then when you get done, it should look like this. So the second line SBatch submits it to Slurm. And then Slurm's going to read those SBatch parameters and look for a compute node and then put your job on the compute node and run.
Yeah.
Please. Yeah, the mouse doesn't work. Say Control-C. And then tab, space, and hit tab. It just prints the content to the screen so you can see it. Okay, now do SBatch space. RU and hit tab. Enter. Okay, now it says submit the job. You can see there's a job ID. Now type in the letter Q and hit enter. Enter. Yeah. So see, it shows your job is running. This is a FastQC job. It's doing the quality assessment on Illumina reads. It's probably done already. Hit Q again. Q. Enter. See, it's already completed. So you should see these files. Okay, well now there's 2 zip files that just has all the images in there. So let's unzip the R1 file, the first read. So you type in unzip DR34 R1 is bottom command. And if you hit DR34, R1 tab, zip tab. If you hit tab, it kind of completes until there's like duplicate entries, and then so unzip DR34 R1 FastQC. That's it. Okay, enter. Unzip. Oh, I don't have unzip. Yeah.
See, solve the calculation. Solve that. DLS. Oh, now it's there is do S batch. S. Yeah,
Yeah, the second one there.
do S batch, just like an S batch.
Oh, I'm sorry. Since we're working on Grace, you need to do space.
Now do S batch. Space. Or use. Something makes out, in the tab, but it'll compare state. And enter. See, this one's called launch. I got that.
Now it's running. It's going to take just a minute. I didn't realize you don't have the unzip command installed. Okay, so let's I don't even know if we don't have links to talk. Okay, we'll just keep moving along, but basically we're going to get this command installed later. There's a links command, and you can see the HTML file. See, we have these HTML files. These are the report files. So in the report file, it's going to show you some statistics that it reviewed, and based on what it knows, it'll let you know whether it passes or fails. You can't type this in right now, and the portal doesn't let you view HTML files anymore. So we'll skip that for now, but it shows you see how this passed, but now it's saying this one failed, per base sequence content failed, and Kamer content failed. Okay. We need to know why it failed. It may just be for a specific reason. So if you open this image, this isn't installed on launch, but you can't do this on launch. I wonder if you can. Let me just check some right quick. You can probably download it. If you double click this, it will download, and then you can open it. So go to your files, Scratch, and then go. You can unselect show dot files, so we don't see as many dot files. And then go to NGS class, and then you can double click this to download it, the zip file. And when it downloads, you can open it, and there's an image, and then you can view images, hopefully. Okay. So everybody get that? Navigate to the directory, Scratch NGS class, and open the zip. Double click the zip, and it will download it for you. Then you can see the images. Okay, let's take a 10-minute break. We'll come back, a 9-minute break. We'll come back at 2:55. I'm going to see if I can get this installed right quick. So let's go back to our files and refresh your page. And then you'll see this directory right here. Double click that, or maybe just single click. One click. Then you'll see a directory called images. Click that. And we want to look at per base quality. So click these three dots and click view. Okay. Per base quality PNG. I imagine SVG is the same. No, SVG downloads. Don't click SVG. Hey, do y'all have this up?
For ESG. Yeah.
Run it. Just go in here, click scratch, click them. You go to NGS class. And then here's the directory with that ones. And then images. And then go down right here for per base quality. PNG, click those three dots. Click view. Okay. So this is just showing you the read length here goes up to 250. So it sequenced 250 base pairs. The green is the highest quality based on the scores. You can see the quality is very good at the beginning, and then towards the end it starts to get not so good. That's when the reagents wear off and the accuracy gets a little bit lower. So there's some of the reads are down here in the red. Okay. So this is what the FastQ file looks like. It's taking the very first sequence. This is actually it's taking the average of the first sequence, the average in the first column, the first sequence, and giving you a quality range here. So this is the top fourth of all the values. This is a middle top fourth, second, or the upper, right above the median, and then lower quartile. Okay, you can see these are four. This is a fourth, this is a fourth, this is a fourth, this is a fourth. So the red line is the median. So the same number above counts and below. The blue is the average. So you can get an idea of so after a while this starts to put in bins. Notice it starts from 1 to 9, then it starts from 20 to 24. So it starts to show you from 20 to 24 is going to be 1 bar. So it starts to bend. So the graph's not so long. Okay. So if you look at per base sequence content, you're going to see something like this. This is the one that failed. So we want it to look, we expect it to look like this, where the A is green, the T is red. So we expect A and T to be the same percent, right? Because they're complementary, right? And we expect G and C to be the same amount since they're complementary. So A and T are at 30% each, so that's 60% total. G and C are at 20 each, so that's 40% total. So that's 100%. So this is what we're seeing. There's a higher AT content than GC in this genome. At the beginning, it's all scrambled like this, but it looks looks almost symmetrical. There's something going on there. FastQC flagged this as failing, but it's actually not failing because the way that the library was created, it uses a transposon insertion site to cut the DNA instead of fragmenting it manually. Same way with the camera profile, it failed because of this insertion site. The insertion site, it is part of the genome, but it seems it has a similar motif because that's what the transposon recognizes in the genome. All right, there's another image called flow cell quality. The flow cell is what you plug into the sequencer. It looks like this. It's a little bit smaller than that. It's about the size of a USB drive. Plug it in. This is MySeq. And then it reads across there. There's a grid, and if it's blue, then everything's good. That means if there weren't any problems. And there's a top and bottom part. if you divide the image in half. And you can see that little streak, light blue, huh?
Question.
Yeah.
Illumina transposon insertion site. If we go back to the
Yeah.
this picture. For the first one, can we use this data like the
Yeah. So you can use that data. It's part of the genome, but read the genome assembler. It may ask you to trim that off because it might do something in the background that says I'm gonna take the first part of the read and I'm gonna assume the rest is similar. And then I'm gonna go with that. So it may ask you to trim it, but you don't have to unless the tool tells you to trim it. But it is part of the genome.
so like here but what I want to say that the next part is kind of giving accurate result. Does this mean and the first one is not giving us accurate result? The rest of it is perfect.
The first part is just skewed towards having higher T and A compared to the rest. That's just because transposon recognizes a motif. This is the pattern of the motif it recognizes in the genome. Once it finds that motif, it lands there. It's not an exact sequence. It's a kind of a flexible sequence. It can either match this or this. When it finds it, it lands there and then it cuts the DNA. So that motif is part of the genome, but it based on this output, it's not as consistent as the rest. Okay, what does a failed example look like? This looks bad. It's just a lot in the red, a lot in the orange. This is a sequence kit that expired. We ran it anyway, just see what would happen. You probably won't get something like this from the sequence center. Here's another one. The fragment content, the camera content, or at the end, there's a big peak here, there's a spike here in the middle, and at the beginning. It should look like this the whole way, right here. Right in there, it should be kind of low. What happened here was the DNA prep wasn't complete in washing off some of the adapters. So towards the end, you see a lot of this on there. They redid the library and they resequenced and it looked better the next time. Here's the flow cell. It should be all blue, but if you see a dot like this, something may be wrong. This basically we wrote to Illumina and showed them this, and they said that is a faulty flow cell. They sent us a new one. Now this is showing you contamination. You expect a GC content to have this bell curve like this blue one, but what you actually see is this red. You see a peak here, you see another peak here, another peak here. So you've got like multiple genomes sequenced. Probably you have this organism you sequenced, and this might be the person working in the lab that sneezed or something, or didn't wash their hands. They got their DNA in there, and then maybe some insects got in there too. So, but if you were to align this to like the NR database or whatever NT, then you would see some might align to human, some might align to your target organism, and some might align to mouse or something. All right. Quality trimming. Now we want to trim these reads that we saw. There's a couple of tools, Trim-O-Matic, Trim Galore. We're just going to go ahead and get into it. So let's go back to command line and type in GCA templates. You can close these windows that were opened for viewing. Let's go back here. You should be in NGS class. You should see these files. So type in GCA templates. This time we're going to use Trim Galore. So just do S for search, type in S and hit enter, and then type in Galore. And notice it took us kind of quickly to Trim Galore. Now you can type one, and then Y for yes. Okay. See how we did that? Everyone get that? Did you get it? Okay, GCA templates.
Yes.
hit S to search, hit enter, and then type in galore. And it's going to search for anything that has the word galore in it. And there it found trim galore. So then you can type in one, and then do you want to copy it? You can type in yes, but since I already have it copied, I'm going to hit Q to quit.
Yes.
Yeah. So if you do LHL, and you should see that script at the top. Remember, LHL prints the newest file to the oldest. If you just did it like this, then you're like searching for it. You're not sure. It's a lot better if you sort it. Okay. Has everyone got this file now? Okay, then go ahead and trim galore. Okay, let's cat that to this. If you do cat run trim galore, you notice that it's configured to use the same input file that we did the first time in the first script, so you don't need to change it. So you can go ahead and just run it. It's using the same input files. It's going to trim those input files and save the trim files to your directory. So type in SBA, hit tab, RUN, hit tab. Now it doesn't complete because there's two scripts. You hit tab a couple times, it shows you there's two scripts. We want to do the trim galore, so hit T and hit tab. So you should do SBatch, run trim galore. Make use of the tab key so you're not typing in dots and dashes. And then you can hit enter, and then you can hit Q again to see it's running. Submit it to the SARM slider. It says SBatch, Q, and it should show running or pending. It only takes a minute or two to run. So did everyone submit it? Who did not submit it yet? You got it submitted? Okay. Okay, see, there's two file options. Which file do you want? You want to do the trim? So type in the letter T and hit tab. And then I'll see. Yeah. Now see, it just went into the screen. Now it's using the same input files as the other script. So now you want to submit it. Just type in SBatch. Space RU tab, and then hit T, tab, C, and then hit enter. And then type in the letter Q and hit tab. Hit enter. So it shows it's pending. You can do up arrow. If you do up arrow, it's your last command and hit enter. And now it shows it's running. It's only going to take a minute or two. Mine's probably already done. Yeah, mine is done. So LHL, and you should see a directory called trim file. Or you know. Okay. Just go backspace. Go backspace. Keep going all the way. Take all that off. All the way back. I'll take all the mark off. And then type in Y for yes and hit enter. Okay, now do LHL. Okay, there's your new script. Trim galore. So you want to submit it to the job scheduler. Remember how to do that. SBatch. RU. Hit tab. Okay, now hit tab. Now saying there's two files. Which one do you want to do? So type in T. Hit tab. Okay, so now hit enter. That's submits it to the scheduler. There's the job ID. So hit type in Q. Hit enter. It shows it's running. It's going to take a minute to complete. And while that's running, we'll move on. Okay, so what we're seeing now, we're trimming the reads. based on quality, or maybe there's an adapter on the end. So what's going to happen if you look at the top two reads from a double-stranded DNA, it's going to look at these, it's going to trim. Maybe the first one didn't have any issues. It's going to keep all 100 bases. Maybe the second one had an adapter, or maybe it was really poor quality, so it trimmed off 50 bases. It's going to save these into the trim files, read 1 and read 2, into 2 separate files. Sometimes the second read is too short. Say you want a minimum of 40. Second read gets trimmed all the way to 20 bases. You don't want to use that. You discard it, but you can keep this first one. The first one gets thrown into another file called single end reads. It means there's not a pair. The pair was either thrown away or it was sequenced as a single end read. There's also a chance that this fragment too, that it overlapped, but meaning the fragment is so short that whenever the sequencer sequenced, it sequenced top and bottom, and the sequence run, it overlaps. So there's a section it sequenced twice in the middle. So now we know this. We know the sequence of the entire strand because they overlap. The top one, we don't know the sequence of the entire strand because there's a gap in the middle. So the bottom one here, the second one, if you can merge them into one long single end read, that might cut down on some computational work, and some you can read the software you're using to see if it recommends merging reads. There's some tools you can use to merge the reads: Flash, BBMap, and Pair. All right, so our job should be finished. So let's do ls trim files, ls tri, and hit tab. And you should see that. So what do you think the format of the trim files is going to be? Is it going to be .zip, .html, or .gz? And do you know what .gz means? Anyone know what .gz means? GZ means is compressed by a tool called gzip. Gzip, whenever something is compressed, it takes it and it compresses, kind of like when you zip a file in Windows, make it smaller. Basically the output is compressed so you don't take up so much disk space. Because a lot of the utilities can read a zip file, you can just unzip it as it reads it. That way you can save on disk space. So the .gz is a zip file, is zipped with a utility called gzip. Now the one that ends in .zip, that zip with a utility called unzip. Notice this one's called FQ. It's FastQ. Instead of spelling out FastQ, it just says FastQ. Okay. So unzip. Now we want to unzip trim files, R1, val1, FastQ zip. Okay. So unzip trim files, R1, val1, .fastq.gz. Oh, I'm sorry, we're doing FastQ. No, we're not doing that. Don't do that. R1, val1, underscore, FastQ, zip. Okay, that's what we want. Could have copied and pasted so there wasn't any. So when you do that, you're going to see kind of what you saw last time. And if you do LHL, you can do LHL trim files. Oh, sorry. I'm gonna do Control L to clear. It's right here. This is the val one. It's a directory. It created a directory. So now you can do LHL and then highlight this
right click, and then you see there's images. Okay, so there's the images we saw. So this is trim galore. It will trim and then it'll run FastQC on the trim reads. So now we can look at the trim reads after trimming, compare it to before trimming and see how it looked. Okay, so did everyone get that? First, you want to unzip. Did everyone get this unzipped? You gonna unzip? Yeah. Okay. No. No. I'll see. Do LS. Enter. Oh, it's there. You did it. Okay, now we're gonna go look at this file back in the browser. So let's go back to the browser. Refresh the page because we have some new files in there. Actually, you need to go back. Go back to NGS class. So you should see this at the top. NGS class. And here's the val1 FastQ that we just unzipped. Okay, back in NGS class, val1 FastQC. Click that, and then click images. And then we're going to do per base quality PNG and view it. You can see it's a little bit better. There's some in the red here, but it looks better than if I go back to NGS class. Here. This is before and that's after. So you can see it cleaned up before, after. See how it cleaned up a little bit? All the values went up. And there's not as much in the red. So if you download data from the web, you'll definitely want to run FastQC before trimming to make sure, because people can load anything up on the web. I mean, somebody might upload this data on the web, right? So you need to know what you're getting into. All right. So everybody got this.
Images.
Yeah, images. So, and then it's fine. Well, actually, here's the very first one. Go back here. It's just gonna tell us like it was like it. So we go ahead and anything.
Right.
Click this three dots. So you can see it's a little bit better on the end. There's only like 5. There's only like 5 in the red compared to the like 8. Okay, so now we did some trimming. Now what I want to do is map these reads to a genome. There's a number of alignment tools depending on the type of sequencing you have. BWA is very popular for aligning to Illumina reads. Parabricks, if you're using a large genome like human or mouse, you'll want to use Parabricks. It'll speed up everything. But this is a fungal genome and it's not very big, so we're just going to use BWA. Bowtie is for RNA-seq data, and we have a lot of genome indexes. So you do need to index the genome when you do an alignment. We already have the index for you, and there's what you can look at the software like BWA to see how to index. It's basically just BWA space index space in the name of the genome FASTA. So we're gonna run GCA templates again. We're gonna look for something that does alignments with BWA. So don't type in 11 since that's gray. So let's search for BWA.
let's go back to, I'm gonna close all these images. We don't need them. Let's go back here. Control U clears it, Control L clears. Okay. GCA templates. Actually, we know we're gonna do sequence alignment. So number 3, we can type in number 3. Align genome sequences to reference genome. That's what we want. Now we have a utility code that uses BWA and SAMTools 1.18. So there's only one option here, and there's only one option, paired in reads. And remember the temp directory, that's the directory on the compute node. It does the sorting, and when it does sorting, it needs to write some temporary file. So it writes it on the compute node. On the compute node drive instead of writing it across the file system to scratch. So that's what that is. So type in 1. Now, pretty long, run, but it's very descriptive. Run BWA version 0717, then run SAMTools. We're going to sort paired in reads in the temp directory. Then Y to save it. Okay. So don't submit it yet because we need to change the input files. We need to use our trimmed reads. So does everyone have a copy of the file? You got a copy? Did you get a copy? Okay. Okay. Let's go page for home. And then type number 3. And then align genome number 1. BWA samples number 1. The options they want to use. And Y to got it. Okay, now I need to go edit this file. So let's go back to the browser. Let's go back to the web browser. You need to refresh this because actually we need to go back to, just go back to NGS class and it might show up. Yeah, there it is. When you click and go back to NGS class, it automatically refreshes for you. Now, remember we were doing LHL. You can also sort by newest. So if you click this, click it again, when the arrow is pointing down, that means you got the newest file on top. Okay, that's that very long file name. Now we want to edit this file. So we want to edit the run BWA file. Y'all get that? NGS class, sort it until the arrow is pointing down. And you have this new one on top, and then you click edit. And you should see this editor. I'll go back. You want to click this, edit. Now when this editor. Okay, now let's go back to the command line because we need to get the names of the trim files. So do LHL-TRI and hit tab. Okay, now do up arrow. There's our command we just typed in. And then we're going to do a wildcard. So do *GZ. We just want to see the files that are GZ. Okay. So you should see these 2 files. These are the trim FastQ files. So now the reason I'm doing this is so we can copy it. See, I copied it. Actually, remember to let's copy 1 first. So copy 1. And then go back to the editor. And line 25, we're going to edit this and we're going to delete all of that. Keep the quotes. And then I think we can hit Control V to paste. Yeah, Control V. And then go back, copy 2. And then come here line 26. Control V. So now we're using our input files for our trimmed reads. Okay, let's let everyone get this first because we still have to do the genome. Did you get those edited? There should be a forward slash or start. Okay. Forward. Enter. Oh, TRI. So you can use the mouse, you use the arrow. Okay, so copy that file on the first one, R1. And do copy some of that, and then go back. And close this. Yeah. And then line 25. Yeah, all that. You just highlight it. And then click Control V to paste. Okay. Do the same for the second. Yeah.
Yeah.
Okay. I got one more thing to do here. Oh, a few more things. Okay. Now we need to set the reference genome. So this line is kind of long. So it's at the bottom here. So what we're going to do is we're going to, first of all, we're going to find the file here. So do ls / scratch, type in scr and hit tab. Data, hit tab. Bio, hit tab. Don't hit enter yet. Let me it's gonna get kind of long. Scratch data bio. Genome indexes. If you hit genome underscore indexes, this one is capital C. Hit tab. Oh, the next one is Gmod genomes. So genome index is Gmod genomes. Capital C, hit tab. And then B, hit tab. BWA. And then capital C and hit tab. And that's the path we want. Hit enter. So all we're saying is list this file. And if I typed it in wrong, it's going to say no such file. I want to make sure I want to make sure this is the correct path before I copy and paste it. So I know it's kind of long, but the index files are already created here in that directory. And this is what you'll do if you're aligning to the human genome. We already have index files for you. I don't know why that happened. Did anyone get terminated just now? Yeah. I don't know what happened there. Okay, hopefully you copied it. I'm going to come back here and I'm going to put it in. If not, I'll go over this again right quick. Ref genome. Yeah, it's really long. But that's what we need. Okay, copy and paste that in there. You might have to start a new session. Just start a new cluster on shell access. I don't know why we got. Okay, now I need to go to CD. Okay.
Get everybody back to where we're.
Uh,
Okay, you need a space after this. Use the arrow keys and go back. Enter. And then go all the way back again. So the Unix command is ls. Now you're going to give tab. Hit tab. Okay. And then B for BWA, B, tab. Okay, then capital C. Hit tab. Okay. Hit enter. Okay, so that you need to copy that line. A very long line here. Just highlight it out. You always. Okay, and then go back. And on line 28, you want to replace that big, yeah. Not the whole line, starting with the scratch. Yeah. And highlight all that. You need to close the quotes. You got two scratch slashes there, so you take one out. And then you can set, and then save that in case you get booted off again. Okay, where were we? File editor. So now what we want to do, I'm going to skip. Okay, I don't want to do. Line 31. It says read group. Let's make the read group just the name of it, DR34. And then you can change the sample name, DR34. Okay. These are paired in reads, so the library stays PE. That's what PE stands for, paired in. So we had to change 5 lines, 25, 26, 28, and then 31, 32. See, all these commands are already ready for you, so you don't have to search and figure out how to run BWA and SAMTools, and save that when you're done. Okay. See, make sure you got everything. Yeah, okay, when you're ready and you saved it, let's go back and submit the job script. So see what I'm doing? SBA tab, RU tab, B tab. Submit that job script. And then hit Q so you can see it's pending. Pending. It's probably going to start running a little bit. But while that's running, I think this one takes maybe 2 minutes. Okay, now it's running.
right now, it's all complicated. What do you need to do?
You need to start a new session. So close that window. Yeah. And go back to actually our cluster launch shell access to it. And I need this remember CD. I'm gonna say this. I've got CD base. Let me tap the command for you. Okay, that's what you need to finish. Everybody got their job running? One more. Wait, no, one more person. Okay, so now we're aligning our trimmed reads to a reference genome. And so what's going to happen, what we're going to see is
we want to find out our sample. The reference genome was built with a known sample. It's done a lot of sequencing, a lot of annotations, but we know where the genes are on this fungal genome. We know where the genes are, the exact coordinates of all the exons on the genome. We're taking a sample, a wild type sample that it's maybe causing someone to be ill, or maybe it's infecting someone. It's hard to kill it with fungal medicines. Now we're finding out what's wrong with this. What's different from the reference genome? What makes this special or worse or non-functional? And what we're going to see is like different base pair changes in compared to the reference genome. A lot of it's going to be the same when it aligns, a lot going to be the same, but there's going to be changes in the coding region. That's what we're interested in, right? Okay, we did that. Okay, so let's just do LHL. Okay, my job is complete. So everyone should see something like this. A DR34 sorted BAM. It's 247 megabytes. Does everybody see that? Now that BAM file it's compressed in a different format. It's not GZ, it's a different format, and you have to use SAMTools to view it. So don't try to cat to the screen or try to open it with your files editor. It's too big, you don't need to go through all that. We have utilities that will help us look at this. So does everybody have a BAM file? Did you get a BAM file? Okay, you submit your jobs with S-Batch, A tab, RUN, tab, and then B for BWA, tab, enter. And type in Q, and then it's running, it just takes a minute. And then when it hit Q again, and when it's done, Q. Okay, it's done already. So type in LHL. You see you have a BAM file. It's sorted. BAM file, 247 megabytes. So now we aligned sequences to a reference genome. Okay, we need to use SAMTools to view the BAM file. What's in the BAM file? We can view a little bit of information. It's interesting. You need to see this. And we're going to do this, and then we're going to go to the next step right quick. We'll have to skip a few slides. So load this module load 1230 SAMTools. Okay. GCC. Okay. So type that first line in. You can type in the second line. SAMTools index. SAMTools will create an index of the BAM file. It helps some software to sort through the BAM file quicker. So go ahead and type these two separate commands in. So you should see a .bai file at the top.
That's a BAM index file. Notice the BAM file, it uses the BAM file name and then .vai. So you know which file was indexed. Okay. Sometimes you might see SAM file. SAM file is the same as a BAM file, except it's not compressed. So it might be like 1 GB SAM file. When you make it to a BAM file, it might only be like 200 megabytes. So it's a lot smaller, save space. And that's the format you want to use BAM files because they can get rather big. You don't need to use up so much space with a SAM file when it can be compressed. Okay, did everyone get these commands run? Not yet. What about over here? You got them? Um, Now you can do model one. Type in SAMTools, SAMTools. Enter. You can type it knows which version. Okay. Now do SAMTools index dr34 underscore student. It's lowercase. The name of the module is uppercase, but when you run the command, it's lowercase. No underscore, it's dr34. And then underscore. Underscore. S, tab. Okay, enter. Okay. And then when that's done, you can do LHL and you'll see like a BAI file. So we're running short on time. So, well, I want to show you this and then we're going to go to doing the variant calling. So you can do SAMTools view. Let's just do the second command, SAMTools view -H, capital H. Okay, that's the one we want to do. We're just going to do the second command for now. You can do the others later, but I want to complete and get to the next script. So when you do H, it shows you the header. Now the very first line shows you that this is an alignment file. It's it's sorted by coordinate. So coordinate, that's genomic coordinate. So these are the names of the chromosomes or the sequence, whatever scaffolds or whatever you're aligning to. In this case, this genome has chromosomes 1 through 7, and then it has a chromosome called R. It shows you chromosome 1 is 3,214,061 base pairs. Generally, the chromosome 1 is the longest chromosome. See, the longest
chromosome is called 1 and they get shorter. And then they've got this R chromosome. So you've got 8 chromosomes. It kind of shows you a history of what's been done, how this file was generated. It shows you that BWA version 0, see, we use 0717. And this shows you the command that was used to create this file. See, it even has the name of the temp directory. The temp directory was created when it ran. And also it used SAMTools. SAMTools version 118. So it has a history of how this was generated, which is very useful because if it didn't have this, you wouldn't know what was used to. Maybe use BWA 0712, and maybe there was an error. There's a bug in 0712, so maybe you need to redo it with 0713, for example. Actually, the index was created with 0712. The actual BWA version is 0717. But the index created with an older BWA version still works with the newer. But let's just say you were using 0717 and there was a bug in that one and you need to redo it. You need to know if this was run with 0717. Okay. And basically, it's showing you what command was used. Okay. So there's a lot of information in this BAM file. I'm going to skip through this. You can read through this on your own. Basically, there's these flags, and it tells you a lot of information about where it's placed in the genome. And so if your flag is 99, the flag is 99 here. It shows chromosome 1 and the genomic coordinate. Then if your flag is 99, then it's the value of 64 plus 32 plus 1. That's 9 plus 2 plus 1. That's 99. So 99 is these 4 values. It's a paired read. The reads are mapped properly. It means they're on the same chromosome. The mate is on the reverse strand, and this is the first one of the pair. So it gives you information about alignment. It can be important. Let's do SAMTools flag stat. Flag stat. 34 unsorted. Okay. SAMTools flag stat, and then the name of the BAM file. This is going to give us some more statistics on percent alignment and if it's aligned correctly. And this is all information you need to review when you're doing your research, because you need to know what's going on besides just running a tool and moving on to the next step. You need to make sure you're getting good alignments, because if you get we hit enter and let that run. Okay. So look at this, percent mapped. Okay, there's 2 values. There's the first value and there's +0. You see a lot of +0s. That's for a second field in the BAM file. We don't have a quality field, so that's why you see +0. So you look at the first value here. There's 1,863,297 reads are properly mapped, which is 98% of all the reads. Now, if you see 1% of the reads align, probably you're aligning to the wrong genome, or else you've aligned a contaminant to a genome you think you're aligning to. So this is a very good alignment. 98% of the reads aligned to the reference genome. Why some reads didn't align? It could be that your sample has the DNA sequence that's not in the reference genome. So it can't align to it because it's not in the reference, but it's still part of your sample. So there's 2% of the reads. Might be a small DNA insert that's not in the reference genome, but it's in your sample. Properly paired means it's on the same chromosome and they're oriented in forward and reverse. If there's one that maps chromosome 1 and one that maps to chromosome 7, they're not properly paired. Okay, that's very useful. We're going to skip viewing this. You can do that later. Let's go to sequence variant calling. Basically, we're going to compare this to the reference genome and see what the difference is. And if it falls in a codon, and we want to see if it changes the amino acid sequence. Sometimes it doesn't change the amino acid sequence. Sometimes it does. sometimes it creates a stop codon, which can stop the protein from being made, and you've got a disrupted protein. It doesn't fold correctly, and it could lead to something wrong. Okay, so the software we're going to use this time is called Freebase. So run GCA templates, and let's search for Freebase. Search Freebase. One, yes. LHL. Now there's my run Freebase version 137 on launch template. Okay, we need to change the input to match our BAM file. So we need to edit this file. So first of all, does everyone have the Freebase script? Did you get the Freebase script? What about you in the back row?
I.
Okay. Type in N for no, and then start it over again. Just type in S for search. Enter. Type in FREE. Hit enter. Let's do one Freebase. All right, I'll press one key to pause. And then you want to copy it, type here. Okay, now do LHL. And see it's on top, Freebase. Okay. Now we need to edit this file. Copy this BAM file right here. DR34 sorted BAM. Copy that file name. And then we're going to go back to our, I don't need that open. I'm going to go back to here. I'm going to go back to NGS class. I need to refresh. And it should show me, I'm going to sort by pointing the arrow down. There's my run Freebase launch, and then edit that file. And then this reference BAM, paste that there. Now we need the reference file correct. Okay, one more thing. Freebase calls variants based on ploidy. So if it's a haploid, there's only one chromosome, one homologous chromosome. Only one, like we're diploid, other organisms are diploid, so they get a chromosome from both parents, so they have two copies of the chromosome. So down here on 26, E. coli is a haploid, so ploidy is 1. We need to make this 2 because the fungal genome we're working with is diploid. So change that to a 2, and change the sample name to DR34. Now click on this until the arrow points down. There it is, Freebase. Now click here and edit. Okay, now line 22. Just the value. That's the variable, so take the value from the slash out of it. You gotta pass. You gotta probably send line numbers. Copy that. Yeah, and of course. Okay, now change the diploid to 2 because this is the diploid. Then change the sequence sample name to DR34. Start with DR. 34. You don't need an underscore. And then the reference genome, reference FASTA. We need to use the same That's in the other script. So let's see. Okay, so let's run this command from the command line grep genome. Okay, let's do this. Sorry. So let's run this command grep. Basically, it searches in the file for something. So type in grep space ref underscore genome space and then the run BWA file. Enter. There's the ref genome right here. So you can copy that. Another thing you can do is you can keep going up arrow and maybe you'll see that long path. Maybe not. I think that when our session crashed, we lost that history. So copy that and then go back to your editor and paste the reference FASTA. Right now it's E. coli, so we need to put that long reference name back in there. Okay. So we had to edit on 21, 22, 25, and 26. Then you can save that and submit it. Okay. You get all the changes made? You're ready. That's everything. Oh, no, 22, yeah. You take it. Yeah, that is one. No, it's this morning. To that, for the university. It's okay, copy that. And go with use and that little non-21 things that's not a specific part of it. And it thumbs up the image. Yes, get 20 games, it's the site.
Yeah.
Listen. Yeah, it's not. You're good to go ahead and say. Literally, good take seconds to do. Did it really steer it really don't? You got 2 quotes for it, 1 out. Okay, save it. So save your file and then let's submit this file. Run Freebase. Okay. Okay, my job is running already. All right, while that's running. So far only 1. Actually, the job's complete already. Nope, something's wrong. I did something wrong, maybe. Uh-oh, I did something. I have a quote somewhere. It's missing, maybe. Maybe the end here. Yep. Yeah, I had a double quote there. I missed a quote, so I have to resubmit it. Okay, so that's running. So now you should see a .vcf file. Does everyone see a .vcf file? If you type in LHL, okay, that's variant call format. It's a different format for variants. It's It's still getting created. See, now it's 3.9 megabytes. Now it's 3.9. Okay. Did everyone get the job running? You did? Did you get the job running? What about your job?
It's crashed.
Space. Are you? FRE. And then Q, enter. It's running. Okay, so it takes a few minutes to run. So we got a minute left, maybe. See what we have here. So Freebase, so when you're calling variants, you can have like during the library generation step, there's a PCR. Sometimes it has a PCR amplification. So basically what it does, you're taking the same DNA fragment and you're amplifying it multiple times. But if you're going to call variants, you don't want to have that same fragment with the same SNP in there counted that many times. So Picard marks duplicates, so you're only counting that once when you're deciding whether that's a variant or not. You'll run into that definitely when you get to variant calling. Variant calling tools, Parabricks uses GPUs, GATK, more for large genomes like the human genome. For small genomes like bacteria, E. coli, or fungal genomes, Freebase is really good. There's other pipelines called VARScan and SAMTools, BCF tools. Here's the BCF format. There's a lot of mapping going on. There's a header up here. It tells you what's in each of the lines. You can look through this and to understand what I've highlighted here. There's an old paper here, 10 years old now. They ran 3 different tools to see which variants are called with each of these tools and see which ones overlap. So Freebase, that's the one we're using, the GATK and SAMTools pipeline. They don't call the same variant. There's some differences from Freebase and these tools, but there's 92% overlap. So it calls a lot of them, but the Freebase may do something that GATK doesn't, and vice versa. So that's why they're calling more variants that aren't found in these other tools. Okay, so if we had time, we could view this in JBrowse, but we don't. But if you were to view it in JBrowse, this is a diploid organism. So remember, we have 2 homologous chromosomes. Chromosome 1, there's 2 copies of chromosome 1. So when you align this, when you look at the alignments in JBrowse, here is you're gonna have 3 things. You're gonna have 4 things. You're gonna have the reference genome at the very top. This is the reference genome. This chromosome is that big. See this red bar? That means we're viewing a region in this red bar in this version here. See how it's kind of expanded. If there's a gene right here, there's a gene we already know about in the reference genome. Here's our sample, we align to it. And here's our VCF file. So the VCF is the variant file. The variant file shows us that in this position, there's a SNP. And the reference, it's a T. So up here and it's a T, but our sample has a G here. So it shows you the variant. It goes from a T to a G, and this is from an A to a G. Now you see these other ones scattered across here? Those are just sequencing errors. Since there's only 1 copy out of all of these reads, it's not going to say this is a SNP. This is a sequence error, so it doesn't call it. So that's why we don't see the variant was called in the VCF file. But here, notice only about half of them have a G, and here all of them have a G. Yeah, all of them have a G. So this variant is on both of the homologous chromosomes of the diploid. This variant here is only on one of the chromosomes. So it inherited the variant from one of the parents, not the other. And they're called heterozygous SNPs and homozygous SNPs. And even JBrowse, you can tell the strand, whether it aligns to the top or bottom strand based on the color. That's what the blue and the red are. Okay, there's another tool called SNP-F. I think our job's complete. Okay, did everyone get a 20 megabyte VCF file? Okay, so that is a file all you could use if you were to view it in JBrowse. It would be this. You would be seeing something like this. It shows you the SNPs. It's just a bunch of text right now. It looks like this, and you really can't open it and read it. You need some way to view it. So that's why JBrowse is good to. We have this on the Grace portal. You can figure out how to do that. So there's after that we want to see, is this SNP in a gene region? And it is in a gene region. See this? There's a gene called CPAR that we already know about. Well, it's in the gene region, but is it in a coding region or is it in a intron? This is a fungal genome. There's not a lot of introns, but it is in the coding region. This bar here is the coding region. Intron is just a single line. Very few introns, but it is in the coding region, and it does change, or it could change the codon sequence. It could change the codon based on whether or not it falls in the coding, whether or not it changes the codon, and the codon changes it to a different amino acid or even a stop codon. So SNP-F will let you know if it's a synonymous mutation, which means the amino acid did not change, or if it's a non-synonymous, which means the amino acid did change. So if the amino acid sequence did change, we want to know, maybe is this in a critical region of this gene? There's another tool called Provian. This is not supported anymore, but it's a really useful tool. There might be newer tools now that use GPUs a lot quicker. Basically, this takes a lot of organisms, it aligns, looks at the conservation of the sequence. If it's in a well-conserved region, which means it's very critical for function of life, and there's a mutation there, chances are it's going to cause some effect on the viability of your sample, or maybe it's making it more infectious or something. So a lower score in this case, a lower score means there's some significance. It's in a conserved region that we don't see in a lot of organisms. But if it's in a non-conserved region, maybe it's in a part of the protein that doesn't matter so much, and we see it in a lot of organisms. So basically, it's doing a search for how well conserved the region is. Okay. Portal apps, kind of went over that. Would have gone through JBrowse, but we're out of time. And you can read about biocontainers if you need to. You can create one, but chances are we can create it for you, or we already have it, or you don't really need it because a lot of the bio software we have installed or it can be installed.